Hi everyone, welcome to my channel Pure Biology. Today I come with another video, that is transcription. Before going to this topic, I just want to say sorry because I was busy for another work, and that's why I didn't upload any video in my channel. Okay, so for that I'm sorry, and obviously I'm very grateful that you are all who subscribe my channel for the growth of this one. Okay, so this is our topic today, that is the transcription. Okay, so before going to the process, the transcription process, we have to learn first important topic not term, you can say term. Okay, so you all know what is a transcription. Transcription meaning that the synthesis of mRNA, clear? Synthesis of mRNA. Next, from the DNA. So this is the actual transcription process, and these are the terms. Before going to the process, we have to learn some terms. The first one, this is the transcription unit. You can see here, this is the transcription unit. What is this transcription unit? It is actually each transcribed segment of DNA. Nothing but that. That means only the DNA segments that are responsible for the transcription. These are known as the transcription unit. Clear? Next, we have to learn that what are called the monocystronic transcription and what is the polycystronic transcription. The monocystronic transcription is that it is seen generally in the eukaryotes, and here it carries the information of one gene. That means here the information of one gene is carried in this transcription process. So that's why it is called a mono. Okay, only one information of the gene. That means the gene, only one gene's information is carried. That's why it is called the monocystronic. Next, we have to learn that polycystronic transcription. What is that polycystronic? In the prokaryotes, it's seen because we all know that the prokaryotes that are very unicellular. So there are the genes information not much more very high. Okay, so you can see in eukaryotes, which are the higher level organisms, they have more informations in the that present in the gene than the prokaryotes. Clear? So here the polycystronic transcription, why it is said here, a set of adjacent genes that is often transcribed as unit. Obviously you can understand that the adjacent, all the genes like that, not all, maximum of the genes that are remain adjacent area, they actually act as unit. Okay, and the transcription is seen in that unit. Okay, so this is called the polycystronic transcription. What is that polycystronic? Again I repeat, here the transcribed, that means a set of adjacent genes information act as unit, and here the transcription is seen in that unit. This is called the polycystronic transcription. Okay, the next one, this is called the primary transcript. First immediate product of the transcription, this is called the primary transcript product. Okay, or the primary transcript. That means the first product of mRNA, you can say, that is produced from the DNA. This is called the primary transcript. Next one, we have to learn the coding strand and the template strand. Okay, coding strand and the template strand. Here, this is a strand. You can see that it is, suppose, DNA strand. Okay, this one, this double helix structure. This is the DNA strand. The first one is the coding strand, and the second one is the template strand. And in case of DNA, we have seen that the two strands are very important for the synthesis of the DNA. But in case of the production of the mRNA, only one strand is sufficient, okay, for the production of the mRNA. And the strand that is responsible for the production of the mRNA is known as a template. You can see here, this is the template. And which one is the coding strand? Here actually not necessary the transcription process to occur here. Okay, so this is called the coding strand, and if you follow it clearly or if you look properly at that, you can see this strand or the base pairs of this strand that is totally similar like this mRNA base pairs. Okay, it is similar. That's why it is said that the transcript is identical with one strand. Okay, this is transcript or the mRNA strand that is identical with one strand of the DNA, and this is called the coding strand. Okay, and you all obviously know in the production of the mRNA or the RNA, the base pair is uracil is important. Okay, uracil is important. So here the A is totally replaced by the uracil, and in the DNA this is the thymine or the thymine. Okay, so this is actually the theme which is known as the coding strand and which is called the template strand. And you can say the coding strand is also the sense strand, and you can say the template strand also the anti-sense strand. Clear? Sense strand and the anti-sense strand. Okay. And now we have to learn which are very responsible for the completion of the mRNA. The first one, this is called the promoter. Okay? The first one, this is called the promoter. Promoter is what? It is just containing of the 6 base pairs and consensus sequence. What is called the consensus sequence? Nothing but the most common base pairs. Okay? Most common base pairs is known as the consensus sequence, and this obviously containing 6 base pairs. Okay, generally in prokaryotes, today I will try to describe the prokaryotic transcription process. Okay? So first we have to learn the prokaryotic promoters. The prokaryotic promoter, this is -10 and -35, and you can see that only 6 base pairs are there. Okay, TAT, AAT, and in -35, TTG, ACA. Okay, so another name of -10, this base pair is the Pribnow box. Okay, this is called the Pribnow box, and generally it is the cis-acting position dependent DNA sequence. Okay, it is the cis-acting, not trans. Cis-acting position. Generally, DNA sequence has the 2 places. One the cis-acting position, and another is the trans-acting position. So you can see the promoter. This remain in the cis-acting position of the DNA or DNA sequence. And generally, it is why it is this promoter is very important, because without promoter, the initiation of the transcription is not possible. This is the second point. And the third point, that the DNA sequence of the promoter region is recognized by the RNA polymers. This is only, sorry for the disturbance, you can adjust that because it's not in my hand. Okay, so this is the RNA polymers. What is the role of the RNA polymers? This RNA polymers actually recognize the promoter region place. Okay, the initiation of the transcription process. Before going to the initiation of the transcription process, the actual promoter region is identified by the RNA pol. Okay, so there is another important role is obviously present in the RNA for the RNA polymers. So this is actual the promoter. Now we have to learn what is the actual role of the RNA pol or RNA polymers. Okay, so now we have to learn what is the role of the RNA polymers. Okay, so actually the RNA polymers, there are 5 subunits. This is alpha, beta, beta dash, omega and sigma. Okay. So, if these are all present within the RNA polymers, then it is called the holoenzyme. Okay, it is called the holoenzyme. And among these 5 subunits, the sigma is the another factor. Okay, so these 4, that is, if not 4, alpha has obviously 2 subunits, alpha 1 and alpha 2. So alpha, beta, beta dash and omega, these are togetherly called the core enzyme. Okay, this is called the core enzyme, and the sigma is the another. This is called the sigma factor. Okay, so this core enzyme and the sigma factor both will remain togetherly. This is called the holoenzyme, and this is actually the RNA polymers. So RNA polymer is generally 2 kinds. Generally 2 kinds means sometimes it is DNA dependent RNA polymers, and sometimes it is called the RNA dependent polymers. So in some viruses, this is called the RNA dependent polymers because there is the template is not DNA. The template is RNA, and during the replication process of this DNA within the host cell, the enzyme So what I am saying that this is the RNA dependent RNA poly. Why this is RNA dependent RNA poly? Because here the template is obviously RNA, not DNA, and generally the viruses that are when they infect to the host cell, okay, in that situation the DNA replication is possible due to the presence of not DNA replication here, because they have the RNA sometimes, okay, like that F2 and R17. Okay, example, these are the 2 examples of the virus. So here the RNA replicases are important, and obviously RNA polymers is important for their replication process or inserting their RNA material within the host cell. Okay, host cell's DNA. When the RNA replicates or the RNA polymers, they actually help the replication process. Then the genetic material that present within the virus's body that insert within the host DNA. Okay, so these are in some, and that situation the RNA is the template. That's why these are called the RNA dependent RNA polymers. Okay, now we learned that the actual role of the factors, that means the sigma factor, the alpha, beta, beta dash, omega. Okay, what are the factors actually, and actually the role of the factors. So alpha factors, generally 2 alphas are there, 2 alpha 1 and 2. It is required. Why? Because it plays an important role in the promoter recognition. Okay, the promoter recognition, obviously the promoter present in the DNA strand, and this is totally recognized by the alpha factor. And sometimes it also plays another role in the interaction with the RNA pol with the another some regulatory factor. Okay, some another regulatory factors. So what is the actual role of the alpha? First, it helps to recognize the promoter region, and second one, sometimes it helps to interact with the regulatory factor and the RNA polymers. Next one, what's the role of the beta and the beta dash? They actually togetherly make up the catalytic center. Okay, the catalytic center. Actually, you can say that it plays the catalytic activity. Okay, or it helps to speed up the transcription process. Okay, and the beta subunit actually helps in the elongation process. Okay, elongation process. What is the role of the sigma factor? It is concerned specifically with the promoter recognition. So what we learned here, that alpha is responsible for the promoter recognition, but the specification of the promoter region which promoter region is important for the transcription process. This is totally recognized by the sigma factor. What is the role of the omega? This actually facilitates the assembly of the RNA pore and stabilizes them. That means its actual role is that these are the all subunits, and they totally stabilize with each other until the total transcription process is completed. This is totally, that stabilization process is totally done by this omega. That is the 5 subunits they assemble together to complete the transcription process with the help of the RNA polymers. Last one is the terminator. What is the role of the terminator? You can obviously know that what is the role of the terminator. Terminator actually enter during the process for the termination, obviously. Okay. So some base pairs that are very important for the transcription process to complete that situation. That means how is that we know when some base pair that are copied during the transcription process or that are copied within the mRNA base pairs, then the RNA transcription process becomes stopped. Okay, and sometimes the rho factor, these are very important. Why the rho factor is important? Because the rho factor actually helps to stop the transcription process. So in that situation, when the rho factor is needed for the transcription process to complete or to stop, then this is called the rho dependent termination. Okay, rho dependent termination. And when rho factor is not necessary for the completion or termination of the transcription process, then this is called the rho independent termination. Clear? So these are actual the terms that are very important for understanding the transcription process. I think I'm trying properly explain that each and every terms. So you can first read and learn these terms, and then you continue the transcription process. Okay, and I will explain the transcription process in my next video. So you all continue with my channel. Okay, and thank you. And obviously I say that the new ones who come in my channel, so please you subscribe my channel. Okay, thank you.